I'm Calin Deleanu, working with the Institute of Organic and Supramolecular Chemistry in Bucharest, and also with the Institute of Macromolecular Chemistry in Iași, both belonging to the network of the Romanian Academy. Well, I started as a synthetic chemist long time ago, and now I'm doing mainly NMR spectroscopy applied to several fields: organic chemistry, macromolecular sciences, food chemistry, and medical research. If I stay to medical stuff and metabolomics, most important thing is that you get a lot of information in one shot from only one measurement. Other techniques, they might be even a little bit more sensitive, but you have to have several analyses to get so many parameters that you get by NMR. So I think this is the main advantage. IVDr, it's nothing different from a standard NMR, but it's very reproducible. The technology behind it and also the standard operation proceedings are so well developed these days that you can run a spectrum in Romania and then repeat the same sample in Germany and you get exactly the same spectrum. So this is really what's behind IVDr in short terms. Again, staying to the medical field, which is the hottest part of our research these days, I started with urine samples immediately after we installed the first good NMR in '96. We started with diabetes and a little bit of targeted metabolic disorders. That was before IVDr was developed. So that was with a standard chemistry instrument. Afterwards, we had the chance to have in the academy the first IVDr in Romania, a 600 megahertz instrument, very well equipped. And now we expanded from urine also to blood, plasma and serum. And we have some quite nice results in the last couple of years. For instance, for urine samples, we have currently a large screening of inborn errors in a maternity in the neighbor country Republic of Moldova in Chișinău. Natalia Ușurelu is leading the group there, and we started to screen all new births in that maternity. So we pick up these rare diseases. We also have monitored some patients over the years. That would be, let's say, an example of the urine studies. If we look to blood, plasma and serum, an interesting result was with a group from the Norwegian Institute of Public Health and the group of Hubert Dirvan with Birgitte Lindemann and Trine Husøy. And they had a cohort with healthy people exposed to the background of PFAS. These are polyfluorinated compounds, which are good for a lot of things, but they have the problem that they persist. They don't disappear, they are not degraded. And there were been some issues on the health, but it was not quite clear their effect. And we just published a paper showing for the first time that the lipoprofile, which is LDL, HDL, and all these fractions, are changed by the background effect of PFAS. So this is not people exposed to particular concentrations, but at the background level. And we've associated this concentration in the blood with the effect on lipoprofile. So that's where IVDr was the key. And staying with blood, we have another interesting work with the group of Maria Dorobanțu in the emergency hospital in Bucharest. And there, people having a heart attack are immediately admitted to emergency unit. And according to the level of LDL, they are given a medicine to lower this level, which is called statin. When we go to have a blood analysis, we are always asked not to eat because the analysis would not be good. The question is, with these people which are in emergency and they are not conscient, we don't know if they have been eaten recently or not. And the question was to see if the concentration that we prescribe for statin is affected by the fact that they were eating or not recently. And we proved again with this IVDr platform that there is no effect on the food intake for the concentration of LDL. So that's quite good for doctors. They can prescribe this medicine immediately without caring when they have been eating. And staying with that, we have another collaboration which is starting now with the group of Professor Adrian Kovic in Iași. He's with the nephrology clinic and the University of Medicine. And now Bruker just released a method for inflammatory markers. So this is the next step. We don't have it yet installed in our laboratory, but the plan is to start this type of research immediately. So that's the future work. The benefit would be that you could do a screening for several markers in faster way than you would do it normally. Even if you have the impression that some classical analyses are cheaper, in fact, sometimes you need to repeat the analysis for another marker, and then might take a long time, and also it would add up costs. So the advantage with such a platform like the IVDr NMR is that you have in one shot a lot of parameters. For some of them you not even think in the moment you run the experiment, but you can go back to data and pick up another marker 1 week later without calling the patient again into the lab. So that's a major advantage. There are lots of things to explore in life sciences, so I have lots of thoughts and we are keen to work on new projects. It's only time that it's the limit. But the dream is to see such instruments not only in our chemistry institutes, but also in clinical environments. So the hope is that, the decision makers will consider also in buying such instruments only for clinical research.